done, passive active transport is done. Now we are concentrating on this passive transport. Okay, so passive transport means it is again are of two types. One is osmosis. Okay, one is osmosis and second one is the diffusion. Osmosis and diffusion, right? So we'll discuss what is osmosis, although we have discussed osmosis a little bit in the previous discussion, and we'll be also discussing the diffusion. Now the diffusion are of, okay, three type. One is known as simple diffusion, okay, simple diffusion, facilitated diffusion, okay, and third one is non-ionic diffusion. So simple diffusion, facilitated diffusion and non-ionic diffusion, right? So first we are going to discuss this osmosis and then we will be coming to diffusion, right? Let's see. So osmosis. Okay. The very simple or typical example of osmosis, which is given in every book, is that if you put one RBC, okay, so suppose this is an RBC, this is one RBC, okay. If you put one RBC inside a solution and this solution osmotic strength is lower than that of the RBC, so this is known as hypotonic solution. Hypotonic solution. Okay. So osmolality, effective osmolality inside the RBC is greater than this solution which is present outside. What happens? All of us know this thing, that water will move from low solute concentration or from low osmotic pressure to high osmotic pressure, from low solute concentration to high solute concentration. So movement of water, movement of solvent from low solute concentration to high solute concentration is known as osmosis. We have discussed the movement of water in the previous chapter also. But here you have to remember that osmotic pressure, okay, is calculated by a formula. Okay, so osmotic pressure is denoted by pi. Okay, pi is equal to G multiplied by C multiplied by gas constant and absolute temperature. So R and T, this part is a constant. R and T, this part is a constant, right? So the G is nothing but number of molecules which is present in the solution. C is the concentration of the molecule present in the solution. So basically, number of molecule, how many numbers are present, and what is the concentration of that molecule. This will give you the osmotic pressure or osmotic strength of a solution. This was first time proposed by Van 't Hoff. That's why this is known as VANT, Van 't Hoff law. So this is known as Van 't Hoff law. Pi is equal to GC multiplied by RT, right? Now, you know that one special type of osmotic pressure is known as oncotic pressure. Oncotic pressure. All of you know this. Oncotic pressure or colloidal osmotic pressure, it is nothing but osmotic pressure due to osmotic pressure exerted due to plasma protein. Okay, plasma protein. So oncotic pressure is nothing but it's a kind of osmotic pressure exerted by plasma protein. If I ask you which protein exert maximum oncotic pressure, your answer will be albumin. What is the reason? Reason is that Because the albumin molecular weight is very, very small. If I give you 1 gram of albumin, 1 gram of globulin, in which one there will be more number of molecule? Obviously albumin, because molecular weight is smaller. So because the number of molecule is also more, and second thing is that concentration of albumin is also maximum in the plasma. That's why, because of this increased number of molecule as well as the concentration of albumin is maximum, the albumin is the major protein for giving osmotic oncotic pressure to plasma. So this is all about the osmotic force or osmosis, you have to remember for your MCQ purpose. Now the second thing we are going to discuss is the diffusion. Okay, diffusion, right? So I told you 3 types of diffusion you have to remember: simple diffusion, facilitated, and non-ionic. First discuss the simple and facilitated. Okay, so this is one semi-permeable membrane. Okay, plasma membrane or semi-permeable membrane. On one side of this semi-permeable membrane, suppose one substance is present which is huge in concentration. So the concentration of these substances is very, very high here. And on other side, the concentration is low. So this is compartment A, this is compartment B, right? What happens? If this molecule, which is present on side A, is a non-polar molecule, or if this molecule is a very small molecule, right? It will move from high solute concentration to low solute concentration without the help of energy. Getting my point? So high to low concentration transport, high to low concentration transport, that is the downhill transport without any energy, is nothing but this is known as simple diffusion. This is known as simple diffusion, right? Okay, so this is simple diffusion. So simple diffusion means one important thing I have to simple diffusion, right? So it is occurring from high to low concentration. So are you finding out any carrier protein here? Answer is no. Carrier protein is not required for simple type of diffusion, although channel protein may be present. Please note down. Channel protein may be present for simple diffusion. So if I give you an example, it will be more clear. The first thing that we have discussed, suppose this molecule is nothing but carbon dioxide gas. So carbon dioxide is transported from high to low concentration without the help of carrier protein, without the help of channel protein, because it's a gaseous molecule. So this is simple diffusion. But suppose there is presence of sodium ion on one half of this membrane, that is high concentration, and it is moving to low concentration with the help of a channel protein here. So this is not the carrier protein, this is the channel protein. This is the channel protein. Sodium is moving from high to low concentration. This is also a simple diffusion. Simple diffusion. So simple diffusion means no carrier protein will be used. No carrier protein is used. Channel protein may be present. Channel protein may be present. Please note down. May be present. But never carrier protein will be present. Okay, so channel protein may or may not be present. Okay, like for example, carbon dioxide, no carrier protein, no channel protein is required. For sodium, channel protein is required. For potassium, channel protein is required. So that is also part of the simple diffusion. But look at the other things. Suppose on one hand side, I have given huge amount of glucose. Okay. Now for transport of glucose, there is no such channel. But for transport of glucose, we have discussed SGLT is there. SGLT, sodium glucose linkage transport, that is part of the active, forget that. So there is another kind of carrier protein present there on cell membrane dedicated for transport of glucose, and that carrier protein is known as GLUT. GLUT. What is GLUT? GLUT is a carrier protein. It's not a channel protein, it's a carrier protein. So with the help of GLUT, glucose is transported from this high concentration to low concentration without the help of energy. Transport is known as facilitated. This kind of transport is known as facilitated diffusion. So what is required for facilitated diffusion? The carrier protein is required. The carrier protein is required for facilitated diffusion. Am I clear to all of you? Please remember, simple diffusion means without any channel is also possible, with channel is also possible. If I ask you during action potential, sodium is entering, potassium is going out, what kind of transport? That is simple diffusion, because channel is not a carrier protein. But whenever the substance is moving from high to low with the help of a carrier protein, that becomes a facilitated diffusion, right? Now, if I ask you that whenever any transport occurs with the help of carrier protein, what are the three properties it will follow? I told you repeatedly: saturation, stereo specificity, and competition. That way, graph, if you plot a graph like this, okay, so where x-axis is the concentration of the substances and y-axis is the rate of transport. Simple diffusion curve will look like this. Simple diffusion curve will look like this. What is the meaning of this? Meaning is that whenever the concentration of substances is rising on one half of the membrane, okay, here, if you increase this concentration here more and more, what will happen? More and more amount of molecule will be transported on other side. There is no limitation, there is no saturation. So simple diffusion means no saturation. More the concentration of substances, more will be the rate of transport. But in case of facilitated diffusion, carrier protein is required. That's why there will be a saturation, and you will find out a curve like this. So initially, more the concentration of substances, more is the increase in rate of transport. But after a certain point of time, there will be plateau of this curve. This plateau means this is nothing but due to saturation, and this is typical of facilitated diffusion. This is typical of facilitated diffusion. So facilitated diffusion means, if you look at the rate of transport, initially the rate of transport is higher than that of the simple diffusion. Okay, it will transport rapidly. But after certain point of time, there will be a saturation, there will be a plateau of transport. And the rate of transport where it is getting saturated, that is a maximum rate of transport that can be done by facilitated diffusion. So this point, this is the maximum rate of transport that can be done by a facilitated diffusion. This rate is known as transport maximum, or TM. Okay, so this is known as transport maximum of facilitated diffusion. That is the maximum rate of transport that can be done by a particular type of carrier protein, that is transport maximum. This transport maximum will be discussed in detail in renal physiology. Okay, whenever we will discuss the glucose transport, we will be discussing there in detail. Right? So for the timing, you remember the difference between this simple and facilitated diffusion. Now, the rate of simple diffusion, okay, rate of simple diffusion is calculated by a law which is known as Fick's law of simple diffusion. Very, very important. Fick's law of simple diffusion. According to Fick's law, okay, the rate of diffusion denoted by letter J, the rate of simple diffusion is equal to d A multiplied by delta C, d multiplied by A multiplied by delta C upon your x, where the d is the diffusion constant, diffusion coefficient, so it's a constant, right? A is the surface area of diffusion through which the diffusion is occurring. Suppose you are thinking of alveoli, oxygen carbon dioxide transport. So what is the total surface area of the alveoli? That is the A. Delta C is the concentration difference or concentration gradient between two sides of the membrane. Concentration difference or concentration gradient. Like for example, in the previously A and B side, what is the difference of concentration? And X is the thickness of the membrane. Thickness of the membrane through which the diffusion is occurring. The membrane through which the diffusion is occurring. All these three are the determinant of diffusion. But there is an MCQ in NEET. Which one is the most important determinant of diffusion? Your answer is concentration difference between two sides of membrane. That is the most important determinant of diffusion, right? So this is all about simple and facilitated diffusion. Now the third type of diffusion that is remaining is non-ionic diffusion. Non-ionic diffusion. Okay. The example, non-ionic diffusion occurs at the level of kidney as well as at the level of GIT for the transport of weak acid or weak base. Okay. For the transport of weak acid or weak base, the non-ionic diffusion occurs particularly at the level of kidney as well as in the GIT. Now you try to understand. Okay. So suppose this is the epithelium cell of kidney. Okay. Kidney epithelium cell. Which part of the nephron? The collecting duct cell of nephron. At the level of collecting duct, there is a process by which glutamine generate ammonium ion. Two molecules of ammonium ion will be generated. Along with that, there will be generation of the bicarbonate ion also by a reaction. Now this ammonium, it contains a charge. So this ammonium, if you want to excrete it in the lumen, if you want to excrete in the urine, so you have to secrete this ammonium ion into the lumen, right? Or in the urine. But because it contains a charge, this cannot penetrate the cell membrane. And there is no transporter, there is no channel for ammonium. So what you have to do? You have to divide this ammonium into H plus ion and ammonia gas. Now this collecting duct cell contain transporter for this H plus ion secretion. There are ATPase pump, hydrogen ATPase pump is there, which will secrete the H plus ion into the lumen. This detailed version will be discussed in renal system also, so don't worry. Okay, hydrogen ATPase is there, which is secreting the H plus ion in the lumen. And ammonia, because it's a gaseous molecule, it will easily penetrate the cell membrane by simple diffusion and it will enter in the lumen. Inside the lumen, again, both of them will combine with one another. Okay, so both of them will combine with one another inside the lumen and they will form the ammonium ion again. Now whenever it become a ammonium ion, can this ammonium ion return back to the cell once again? Answer is no. I am asking, is back diffusion of this ammonium ion is possible? Answer is no. So what is the fate of this ammonium ion? This ammonium ion will be excreted at the level of urine. So what is happening? This whole thing, what is happening? So I can understand that diffusion of ammonia is only occurring in non-ionic form. Whenever it becomes an ionized form, the diffusion is prevented. The diffusion is prevented. That means I can comment like this, that the diffusion of ammonia only occurring in non-ionic form. So diffusion only in non-ionic form, not in ionic form. That's why this kind of diffusion is known as non-ionic diffusion. So basically ammonium ion is trapped inside the urine. Okay, so whenever it is non-ionic in the form of ammonia, then diffusion. Ionic form in the form of ammonium ion, no diffusion. That is known as non-ionic diffusion. Getting my point? So this is all about the active transport and passive transport. Now this diagram I have kept here from Guyton, so that it will give you a summary of the whole transport process that we have discussed so far. Okay, what I told, simple diffusion. Okay, either it can happen directly through membrane or it can happen with the help of channel. Look at here, exactly the same thing. This is a channel protein. Okay, so with the help of channel protein or without the help of channel protein, if the molecule is transported through cell membrane, like for example carbon dioxide, like for example oxygen, like for example sodium, they are what type of diffusion? They are simple diffusion. Now look at the carrier protein. Carrier protein, I told you already repeatedly that it may require for diffusion also, that is facilitated diffusion, not the simple one, facilitated diffusion, or it may be required for active transport. See here, this is a carrier protein. Okay, this is a carrier protein which is attached with the substances which is going to be transported, and it is transporting the substances from one part to other part. All type of GLUT are example of this kind of transport. This is facilitated diffusion. Carrier protein can also use energy, and if the carrier protein is utilizing energy, then they will be known as the active transport. Maybe primary active, maybe secondary active, whatever. So this active transport as well as facilitated diffusion, they require the help of carrier protein. But simple diffusion never require the help of carrier protein. It may require channel protein. This is given in Guyton, so this will make you more clear. This will make your concept more clear, right? So now we are left with this process, which is known as endocytosis and exocytosis. So endocytosis, large molecule will enter inside the cell. Simple. All of you know from your class 2 level knowledge that endocytosis are of three types. One is phagocytosis, that is cell eating. Pinocytosis, that is cell drinking. The fluid, the liquid is entering in the cell. And the special one that you have to remember for your MCQ is clathrin mediated endocytosis, which is also known as receptor mediated endocytosis. You remember this word, okay? This will give you answer in MCQ. Receptor mediated endocytosis, that is clathrin mediated endocytosis. So whenever I am saying this clathrin mediated endocytosis, question is, what is clathrin? So clathrin is a triskelion protein with 3 limb, right? So this is 1, this is 2, this is 3. So it is a triskelion protein, okay, which is known as clathrin, it is composed of 3 heavy chain and 3 light chain. You can find out. So this is the heavy chain and this is light chain. What is the role of this clathrin protein? It is a protein which causes coating on vesicle. It gives a coat on vesicle. So here you are finding out this is a vesicle. Onto the surface of this vesicle there is a coating, and this coating is given by this clathrin protein. Clathrin required the help of another protein which is known as dynamin, dynamin, which is present at the neck region of this vesicle, and on the surface of this vesicle the clathrin protein itself is giving the coating. Now how this is happening? How this is happening? Step by step fashion, right? So first of all, suppose this is a cell membrane, okay? On this cell membrane, huge amount of receptor is there. This receptor may be receptor for hormone, this receptor may be a receptor for any kind of ligand. But suppose huge amount of receptor is there. I have to down-regulate this receptor. Down-regulate means I have to take some of the receptor from membrane to the inside the cell so that the effect of the ligand is decreased. That is known as a down-regulation. Effect is decreased, down-regulation. How to do that? Whenever ligand is attached with this receptor, suppose now the receptor is attached with the ligand. So this is receptor ligand complex. Whenever this receptor ligand complex occurs, what will happen? This receptor will aggregate at a narrow area on the membrane. Aggregation of the receptor along with ligand is happening, and then this will be coated by this clathrin protein. This will be coated by clathrin. This is not actually coating. This is known as recruitment of clathrin. Recruitment of clathrin protein. Then after the recruitment of clathrin protein, what will happen? There will be appearance of a depression, invagination of this membrane. There will be appearance of an invagination of this membrane. At the level of this invagination, all these receptor is there. All these receptors are there along with their attached ligand, right? And this is already coated by clathrin. Already coated by clathrin. Now next is that this depression area, now it will be detached from the membrane. It will form a pit, and that will be detached from the membrane as a vesicle. And this vesicle has coating of clathrin on the surface that we have seen already. So you can imagine like this, that this is the vesicle. Inside this vesicle the receptor along with the ligand is present, and surrounding this vesicle the clathrin protein is coated. Now the next thing is that now there will be uncoating of clathrin. Uncoating of clathrin, and the clathrin will be reused here. Clathrin will be recycled here. And what is the fate of this vesicle? The vesicle will fuse with lysosome. The vesicle will fuse with lysosome, and all of you know lysosomal enzyme will causes degradation of this receptor, and this way the down regulation of receptor is occurring. This is known as clathrin mediated endocytosis. So basically clathrin is helping in endocytosis. What endocytosis? This is typically occur for receptor endocytosis, and that's why this is known as clathrin mediated endocytosis, right? So this is all about different types of endocytosis. And the last one is exocytosis. So exocytosis means if this is your cell, suppose this is a large cell, okay, suppose this is a large cell. Inside this cell, the protein synthesizing organelle is rough endoplasmic reticulum, okay. So suppose rough endoplasmic reticulum is synthesizing various proteins. This protein can be pump, this protein can be carrier protein, transporter protein, anything. But you all know that if this protein is going to be secreted from this cell, what you have to do? You have to pack this protein inside the vesicle. Otherwise, the protein cannot be transported alone. You have to pack this protein inside the vesicle. And the organelle, which causes packaging of the protein, we all know, okay, that is nothing but Golgi body. Okay, so this protein molecule is going to enter inside the Golgi body. The side from where it is entering is known as cis side of Golgi body, and the protein will come out from the opposite side, that is known as trans side of Golgi body. Now when the proteins are coming out, it is already packed inside the vesicle. That's why we say Golgi body is responsible for packaging into vesicles. Now, what is the fate of this vesicle? Now this vesicle will move inside the cell by intracellular transport process. We will be discussing this in neuron. After transport, some of the vesicle may directly secreted out of the cell. Okay, so I'm writing this is direct secretion. Direct secretion. This kind of exocytosis is known as constitutive exocytosis. Constitutive exocytosis, right? This kind of secretion, this kind of exocytosis is known as constitutive. Always the secretion is going on. Whenever the vesicle is formed, the secretion is going on. But in the second type, in the second type, these vesicles will reach to a certain places of the cell where it will be stored for some more time. Okay, so what I'm saying, these vesicles will move to a particular portion of the cell where there will be temporary storage of this vesicle, and during this temporary storage there will be pre-processing of this vesicle. Storage is also going on and processing is also going on. Okay, so more and more refined molecule will be produced, and then only the vesicle is going to be secreted. This is known as non-constitutive secretion, okay, or non-constitutive exocytosis. Sometimes it is said that non-constitutive secretion is a regulated exocytosis, and constitutive is a non-regulated exocytosis. But that is not true. That is basically a misnomer. Both of this secretion, whether it is constitutively secreted or it is non-constitutively secreted, both of them are regulated by cell. But when the vesicles are stored in certain area and pre-processing is done, that is more regulated. So that's why sometimes it is also known as regulated secretion, and non-constitutive one is the non-regulated secretion. But that is not absolutely true, right? So this is all about exocytosis. So exocytosis are basically of two types, constitutive and non-constitutive. At the end, we will be discussing certain MCQ. So first one, receptor mediated endocytosis. Very easy. It requires clathrin. Binding site present on beta subunit. Beta is the binding subunit, I told you, and alpha is the all regulatory. So binding subunit beta contain glycosylation site, 3 glycosylation sites. So this is the answer, right? Next, active transport across cell membrane is mediated by, so it is active, okay, across cell membrane. So immediately it has a tendency that it will answer this sodium potassium ATPase pump. Sodium potassium ATPase pump is an also active, but that is a primary active. But if I ask you secondary active, who helps secondary active transport? Carrier protein. Who helps primary active transport? That is also carrier protein. So basically, if we think of primary active as well as secondary active transport, in both the cases, the protein which is required is known as carrier protein. So better answer of this question is carrier protein, although second answer that is possible is sodium potassium ATPase pump, because you have to consider both active transport as well as primary active transport as well as secondary active transport. Now the last question. Solution A and B, they are separated by a membrane. So this is a membrane, solution A, solution B, right? Urea is present, so urea is easily permeable. Solution A contain 10 millimole of urea, and solution B contain 5 millimole of urea. So what will be the rate of diffusion? It depends on delta C, the concentration gradient. So the concentration gradient is 10 minus 5, so it is happening 5, okay. So the rate is 5, number 5, okay, maybe in millimole per minute or millimole per second, whatever. Now the question is saying that if concentration of the urea in solution A is double without changing B, so now you are changing this concentration 10 to 20, that is a double, keeping the same concentration on compartment B. Now in these cases, if I calculate rate, again it will depends on delta C. In these cases, delta C is 20 minus 5. 20 minus 5 is 15. So previously the rate was 5 millimole per minute or second. Now it is 15 millimole per seconds or minute. So it has changed 3 times. That's why the answer is triple. Getting my point? Although it is looking simple, that if you make it double, the transport should be double, but you have to check the delta C, how much delta C, keeping the B concentration same, right? So this way you have to solve MCQ.